the title here, The Case for Monitoring Allergen Content of Food Extracts. I mean, what we're going to talk about are not only extracts, but with food, it's such a huge field of products. So we're going to talk about diagnostics, therapeutics, vaccines, and also some kinds of other foods that are out there that are being used in the allergy space at the moment. So these are my conflicts. A lot of the things I'm going to talk about are products that our company produces. We have our own RO1 to Anapolmaeus, which is actually now in its 14th year, which we're very pleased about. And some of this work was supported by the European Union. So we got into this in 2017. This report, which I'm sure many of you are aware of, was a 3-year-old boy who died during oral food challenge. It was the first death, according to a double-blind placebo-controlled food challenge that we're aware of. And at the time, we had developed a couple of assays for milk, BOSD5 and BOSD11. And so I said, the folks in the UK had the assay. I said, why don't we do some studies looking at what are the allergen levels in milk, and how does that change when you make muffins and everything? So we devised this study where we made muffins according to the Mount Sinai recipe, and we also used the NHS recipe as well. But when you do that, if you look at what happens was the BOSD5 allergen was completely denatured, completely destroyed as a result of the heating process, which was 350 degrees for half an hour. But BOSD11, after this process, the total allergen in the baked muffin was 180 milligrams. And so this allergen, the whole theory of this was based on destroying linear epitopes and everything else. So it was quite clear that these muffins contained a potent amount of allergen. It retained its IgE binding, and you could inhibit the IgE binding to the muffin mix, to uncooked muffin mix by 80%. So really, the baking wasn't doing very much. And actually, when you measure the core temperature of the muffin, it was about 200 degrees. So at the time, also, Hugh Sampson's group had come out with a recommendation that patients with more than 20 units of IgE to BOSD11 were at high risk of failing food challenges. And so this really was confirmed by our data, and we really sort of got us into this whole area of looking at allergens in foods. We've continued to do that with Karina Venter. Now we're doing that with Karina Venter and Alison Hicks at the University of Colorado. And they're looking at allergens, egg and milk allergens in different foods that they're working on, the egg ladder, the milk ladder, and so on. So we then moved on to, well, at that time, as you know, the LEAP study came out. And that was also interesting because in the first year of life, 70% of the children enrolled in LEAP were given BAMBA. And at the time, what is this BAMBA? You know, we hadn't heard of it. I think most other people, had anybody here heard of it at that time? Oh, Larry, of course, had heard of it. In fact, we used to take, so the thing about BAMBA is, number one, you can understand why kids are weaned on BAMBA in Israel. You taste this, it melts in your mouth, it's got a nice flavor, absolutely an ideal weaning tool, let's say. So we decided to go ahead. We had a few assays, ELISA assays, to look at what the allergen content in BAMBA was. And so this is the data. We were absolutely surprised by the reproducibility of this material. We looked at 8 samples from the US, 8 samples from the UK, and it was just very close. This is not a log scale, this is just a regular scale, but these numbers were very tight, and there was more or less an equal ratio of these major allergens in the product. And we're comparing that with another product which is called Erdnussflocken in Germany. Has anybody had any of that? Larry? No? Okay. It's given out at adult parties in Germany, and so that you can have peanuts and beer at the same time. Anyway, when we made the allergen measurements, we calculated that roughly 330 milligrams of these allergens was associated with prevention of peanut allergy. And the BAMBA, very well formulated product, and to us, the data was almost like a pharmaceutical. So that was really what we were very, very interested in. So I just want to go a bit about the technology that we've been using. We developed an assay a few years ago called Maria, which is a multiplex array for indoor allergens, and we can measure 8 or 9 allergens at once using this technology. We then developed that assay for foods, and so this uses the Luminex platform, which is the one that typically one uses in immunology for cytokines and other mediators. The idea really is that the beads here that are provided by Luminex, you can couple monoclonal antibodies to those. You can do up to 100 different beads. This is like an ELISA assay, and then you detect it with streptavidin phycoerythrin. You put it through a laser. The red laser reads the beads. The green laser, I think, reads the SAP. And then you can get precise measurements of all of these different allergens. So it's like an ELISA on a bead, but you can sort them all out. And it's a very sensitive, actually, very sensitive assay. And these are all the reagents that we were using in Maria for foods. And so our target allergens were selected based on US regulatory and European regulatory information about what's regulated. We'll get to that in a minute. But the thing about this slide is that every component of this test is standardized or standardizable. So the capture antibody, the detector antibody, we can have an allergen standard that's either recombinant or natural, but it's defined according to amino acid content. We can have different target proteins, and we can have a uniform, different sets of beads. So this is all a standardizable system, and that's why we like it. And then these are the regulated foods in the US and in Europe. The latest one that was added a couple of years ago now was sesame. I think the FDA is, this was obviously because sesame allergy has been increasing. I think the FDA is kind of regretting its decision to do this because they've had so much pushback from folks like McDonald's, Sesame Buns, anybody? Chick-fil-A, all these major corporations that are having to now recognize this particularly important allergen. But that's certainly, our goal with this was to develop a test where we could essentially measure most of these allergens at once. So in doing that, and this is case 3, there's going to be half a dozen of these cases, right? So early introduction foods. So in the aftermath of LEAP, there were a whole set of companies that came up and developed products that could be used instead of BAMBA. And so and a lot of companies got into this. You could make peanut powders, you could make puffs and that kind of thing. And so we, having developed the assay, we then thought, well, let's look at these products and let's see what they contain in terms of allergen content, because these are being promoted all over the web and social media to consumers for, quotes, prevention of food allergy. One company got an actually qualified health claim from the FDA, which basically said, yes, there's sufficient evidence out there to say that this works. Now, the one company did all the work, and then all the other companies jumped in the bandwagon and said, oh, there's a qualified health claim. Let's go with that, right? So these are the companies. And when we did this study, we didn't want to do this so that we didn't want to we wanted people to know what the products were and what the companies were. We didn't want to anonymize everything because that would just be a waste of time, basically. So what we did was we gave them these cute little airport codes of MME or RSF or whatever they were to sort of lighten that up a little bit. But these were the foods that we compared. We had no financial interest in any of this, and we also used BAMBA as a sort of reference material because we know BAMBA works. And so if these other foods work, then they ought to have similar allergen levels to BAMBA. That was our thesis. So we looked at early introduction foods from 9 manufacturers and 4 controls. We looked at 32 different foods altogether. We measured RH2 by ELISA because that has been difficult to adopt on the Maria system. And in total, we looked at over 1,500 data points in this study. So all of the products are listed there. These are the controls that we use. PB2 is a peanut butter powder that's commonly used by allergists for oral immunotherapy, and we'll talk a little bit about that later. Peanut butter here, which is an almond, I think it was peanut-free butter, Erdnusslock, and a peanut butter control there too. So when we did this, the findings were that most of these companies had what we call consistent allergen profiles, that meant they were comparable to BAMBA in their allergen levels. And that was especially true of the peanut powder products. But there was up to 100-fold differences in many of these products. We also detected cross-contamination in some products, which were supposedly might be peanut, but they would have contamination with milk and egg, that kind of thing, and inconsistent profiles in 3 companies. So when we look at that, I'll show you the food puff data. So this is BAMBA, Erdnusslock, and another one, Mission Mighty Me, MME, and SFO. And you can see clearly here that the SFO product contained no allergen. If you look at the others, you can see a relatively consistent profile. The blue there is RH3, but you can see that these are looking at around 1,000 to 10,000, over 10,000 micrograms per gram of allergen. The SFO product was negative, and when we did another test of that by SDS Page, this is what we got. And you can see that the 3 samples on the left there, those samples could almost be replicates from a single company. They're very reproducible, whereas the SFO product was clearly degraded, and that's consistent with the findings that we had, that we're showing there in the graph. So we then looked at some other products. Some had dose responses that you gave a different amount of the product on each day. So there was one there, HP, which was peanut, and you can see a nice dose response there, increasing levels of allergen with increasing as the dose was escalated over an 8-day period. The RSF product was a mixture of milk and egg and peanut powder. And you can see the start off on the first few days there is milk, and then days 5 to 8 you add egg, which is in the green, and you start adding peanut later. And this is also quite interesting, because what you see with this is as they do the mixing, the level of milk goes down and egg goes down, and then the peanut comes up. And so that's, again, fitted with what we might expect from this kind of product. And again, you can see high allergen levels there. Some of those milk allergen levels were in the 40,000 micrograms per gram. So this was, in fact, a demonstration of how one can use this information to potentially see what you've got in the product and potentially improve products that may not be at that level. And this was the variability in allergen levels that we saw in multi-blend early introduction foods and fruit sources. And this basically was kind of all over the place. And the problem here was that some of the products were being marketed as having, quotes, a consistent allergen profile with more or less equal amounts of the allergen, which is clearly not the case. And we put a line in there, 10 micrograms per gram. as being indicative of a trace level of contamination, because that's what we found in the study in some of the products. So we might have a milk or an egg or a peanut level of 1,000 or 10,000 micrograms, and we sometimes see trace levels of milk or egg around that level, 10 micrograms. So okay. So if we looked at the combined doses, and this was basically, we used the manufacturer's recommendation for this. You can see here the different servings, what people were getting in terms of cumulative allergen dose for peanut, and also there for milk and egg from the RSF material, and the SFO material by comparison was, 10-fold lower or more. So this, I think, is just a demonstration of how, if you're going to formulate these products, the allergen levels can be very valuable in determining what's in there and how you can make changes in the process of how you formulate these products to produce something that's better. And we have subsequently been working with one or two of these companies to actually do that. So this was all well and good. And then we come to case number 4. And so, as many of you are aware, in December 2022, the FDA issued this warning. 4 different lots of peanut extract, diagnostic allergenic product, were withdrawn because of deficiencies in the product. And also increased reports of false negative skin tests. And as a result of this, what was happening was people were getting false negative skin tests. They were going out and eating peanuts and having reactions. We don't know too much about that, but that's actually clearly was the case. Now, subsequently, in March of last year, the FDA then put out a notice to say that false negative skin tests should not be assumed to be negative, and that the patient should be warned that they may still have reactions to these extracts, food allergenic products. And their warning turned out to be prophetic, because in September of that year, ALK had to withdraw another product, which was pecan. For the same kind of reasons, and someone had a negative skin test and then had a life-threatening reaction after consuming pecan. So the Academy put out this rather sort of, I don't know, it was sort of a knee-jerk response, I guess I would say, to this. This was put out in March of 2023, concern that this could apply, that it was unnecessary. Well, we've heard that before. That the new label statement could be applied to food-specific serum IgE testing, and there's no grounds for making that statement. Because unless you know what's happening with the food extract in the skin test, how can you say that it applies to what's happening in vitro testing, where the extracts may be different? And then this sort of stuff about over-testing, over-diagnosis. I mean, I think this was just a knee-jerk response, and they said they were going to meet with the FDA and discuss it. I don't know whether they have or not, and where this has gone subsequently. So at the time, John Hemler, who I was talking with us about last night, he had noticed, he was doing some studies with Scott Smith on the IgG4 variants of the IgE monoclonals that Scott has to peanut. And in recruiting patients, he'd noticed that they were getting negative reactions where they thought they should be getting positive reactions based on history. They tested them with the Allergen Greer product, and lo and behold, these patients were good. So they thought there was something going on, and John got together with Cosby Stone at Vanderbilt. And so what they did was they did skin testing. And so you can see here the results of skin testing in the recall and the non-recall lots. And clearly, the non-recalled lots, much higher levels of skin test reactivity, really sort of in some cases barely positive skin tests on these recall materials. I want you to draw your attention to these points here. So these were 2 patients who were completely negative, and when they were tested with the non-recall lot, one had a skin test of 14 millimeters, the other one 36 millimeter wheal. And what we noticed with Scott's data actually on the peanut antibodies was that a lot of these patients were getting 20-meter plus wheels. And certainly from our perspective, I'll show you some more data on this in a second, that is really too hot. I mean, I think that's too potent, and we'll get into that in a second. Anyway, what happened was Scott and Cosby, they ran these in a human IgE monoclonal antibody assay. On this graph, you can see these recall lots are in purple and in red, and you can see these curves are shifted by 10 to 100-fold. 634 extract contained virtually nothing. And so there was clear demonstration with these IgE monoclonal antibodies that there was a problem with this. And we then looked using our immunoassays, mouse ELISA for this, and calculated the values in terms of micrograms. And what you can see here are the values of the recall lots, microgram levels. These are actually the non-recall lots, and you can see they contain significantly larger amounts of allergen. Now, what was also interesting to us was that they contain predominantly RH2 and RH6. And that was a surprise because it was a different, that profile was different to what we had seen in the US in extracts that we prepare. And initially we thought, well, this ALK, maybe they've done some processing of these extracts so that they could actually get enrichment for RH2 and RH6, which you can do relatively simply. So we then looked at other extracts. And this is just an SDS page. So you can guess from this which are the recall extracts, right? Anyone? That's the question you have to put your phone in. You put the answer in your phone and send it to Tom. Okay. So yeah, lanes 5 and 6 were the recall materials, and the lots that highlighted in yellow there, we confirmed the data by mass spec. Saya Agar, who did all this work, one of our main scientists, she was excited when we showed this at the European meeting last year. But definitely, there was no protein in it. So how does a company send out an extract that contains no protein, no peanut protein? We don't know, but we did go and do some more testing. So this was testing of European extracts from a company called Low Pharma in Italy and Allergen. And you can see again that these extracts were predominantly RH2 and RH6. But you look at this concentration here, you're talking about 700 micrograms per ml of allergen in the ALK materials in total. With these Allergen extracts, you're almost at 3 milligrams per ml of allergen to do skin testing with. This is crazy. And so it emphasizes the need for further standardization of these materials. If you look, we compared those with our other peanut products down here. This is a material that we manufacture, and it contains sort of more or less, it contains a lot of some RH3, RH1, RH2. This is BAMBA here, and it contains a lot more RH3, and also the PB2, roughly equivalent amounts of the other allergens, but tenfold more RH3. So it just shows how when you're looking at these different materials, how the allergen content can vary. And this is just coincidentally, this is a correlation. We also set up some IgE assays with this. This is a comparison. They were highly correlated with the mouse ELISA. And so really, in terms of allergen standardization, it's amazing that we've been 20 years now in food allergy, and neither the FDA nor the European authorities have produced any kind of food allergen standard. It's really kind of, it's just not good. This is what we think is the approach. You can do biochemical testing. We've shown some of that today. You can run immunoassays based on standardized proteins. You can do mass spectrometry if you don't have an immunoassay. These human IgE monoclonal antibodies from allergic patients can also be part of that equation. You can develop customized methods for non-novel therapeutic products. So these might be allergens that are incorporated into virus particles, for example. And the key thing here is that the standardization is based on measuring allergens, and the allergens are the active pharmaceutical ingredients at the end of the day. And that's what we want to measure. So if you're a drug company, you measure the API of your small molecule. If you're an allergen company, this is what you should be measuring. Okay, just a few examples. There are some reference materials out there from NIST. I mentioned some yesterday. NIST peanut butter is up here. And then there's another organization called MoniQA. We ran these in our Maria for Foods assay, and you can see that you can measure precisely the major allergens in these. You could actually run studies. You could run a ring trial with NIST peanut butter and say, let's hone in on that, and let's look at that content. And that could be a standard for this. But mass spec, there's a lot of excitement, a lot of talk about using mass spec for standardization. We use it now routinely for monitoring the purity of the allergens that we manufacture, for purity assessment of allergen molecules, for quantification, although that's actually quite difficult. But the relative abundance is something that you can do, and it's already being considered by the FDA and the Paul Ehrlich Institute. I wanted to show you some recent data from Sabina Wunschmann in our group. This is 5 years of stability testing of that peanut protein mixture that I showed you, and it maintains its stability over that time period. And we recently produced some others. We produced a reference materials for hazelnut and soy. We're working on others. But for these, we use mass spec, and you can see the mass spec data there for the hazelnut allergens, and here also for the mainly gliam-5 and gliam-6 in soy flour protein. So these are the ways in which we can produce standard reference materials and validate them. So in the final few minutes, I just want to talk about therapeutics a little bit. So how many of you in the audience do oral immunotherapy? And how many of you use Palforzia? One person. Yeah. And how many of you do your own formulation of things? Right. So what do you guys use?
I've been using the bird mill, but they just stopped making it, but I still have leftover with that.
Okay, you can actually get it from Golden Peanut because the bird milk came from Golden Peanut. Okay. But what about the back? What are you using? Peanut butter. Okay, okay. So this is all good. So what Palforzia did, what Aimmune did was they looked at a whole lot of lots of golden peanut flour, and they screened them using these different assays. They looked at allergen content, potency, aflatoxin, a whole number of different things. And when they did that, according to their acceptance criteria, up to 60% of this source material was rejected because it didn't qualify under one or two of these criteria. So when we look at allergen levels, the allergen levels in the studies that they did were really quite tight. And you can see here that the major allergen levels, RH1, 2, and 6, this is relative potency. They varied by about twofold. And that was consistent of this product. So you know then that if you're comparing the data in the Palisade study with the Artemis study, you've more or less got the same allergen concentration. Now, although I tried to encourage them to say, well, how much is it in micrograms, they really wouldn't go for that. So I'm sure that Brian probably knows, but we'll see. Anyway, so the question then is, what's happening out there in the real world? Well, the real world has been this. This was Wasserman's group in Texas, and they published this paper a few years ago now, but they had looked at oral immunotherapy in their clinic. And although 79% of the patients achieved a maintenance dose for over several years, 2,000 milligrams, there was a very high percentage of these patients that were getting adverse reactions that required epinephrine or had other types of adverse reactions related to oral immunotherapy syndromes. And these are the doses that were associated with that. You can see that there's a huge range of doses triggering these adverse events. So what's going on in the real world? Well, this is what's going on in the real world. This is Captain Crunch and the Kool-Aid. And so this is the latest rock band coming out of the allergy community, by the way. And so these are a couple of different protocols. The Captain Crunch protocol, they have an escalation day. They have 1/4 to 2 pieces of this. They build up they have updosing to get to 16 to 120 milligrams. Then they slip in PB2 in teaspoon amounts that get you up to 2,000 milligrams. And then for sometimes they use peanut butter. Okay, so this is one protocol. This is the actual real-world protocol that the Wasserman group is using. And they extract their peanut in distilled water. We don't know what the effects of that are. They have cereal dilutions down to 2 micrograms per ml in distilled water. 11 doses dissolved in Kool-Aid powder. And you can get choice of grape or strawberry or raspberry, whatever. And then the updosing for that is equally, doses of peanut fragments or peanuts, challenging with 24 peanuts at the end, and then putting them on 2,000 milligrams of maintenance dose for 3 years. Now, what I'm saying here is that it's okay for you to do this in your clinic, but the question is, what happens with patients who are crossing state lines? What happens? How do you measure that a teaspoon is uniform from one nurse to another? Those kinds of issues come up, and when you're looking at adverse reaction rates of over 20%, then this is not good. So I'm going to move on from this and just say that the case really for monitoring the allergen content of food is really closed. I mean, we need to make these measurements if we're going to improve food allergy products, for looking at the profiles, for looking at the active pharmaceutical ingredients, for looking at whether we can get tolerogenicity on these early introduction foods. I think at this point it's a must for clinical trials. How can you do a clinical trial where you don't actually know the dose of the active ingredient that you're injecting, that you're administering? And for quality control and management, it's also important. So I think the positive thing about this is all the tools are there. We can do this, as part of the World Health Organization Allergen Standardization Committee last year, they made a commitment to at least standardize peanut. Now, that may take another 5 years, may take another 10 years, I'm not sure, but we'll get there in the end. So I'd like to just acknowledge our collaborators with this, particularly John and Karen Braden at UVA, Scott Smith, Cosby Stone, all of our folks that did this work both in the US and the UK. This is our group at that time in the Common House downtown, which is where all the entrepreneurs hang out, you know. Thank you very much.
The samples that were coming from, that were the peanut failed samples. And they said, actually, their protocol didn't include a protein measurement. Mm. And so they didn't have to check it. And all there was is a protocol that had been accepted at some time from the company, and it says peanut added at this point. And they couldn't, at that time, couldn't say what had gone wrong.
Mm-hmm. Yeah, I mean, it is remarkable, and especially from ALK, because the archetype of ALK is that we are the allergen manufacturer that, you know, monitors everything in our products and so on and so forth. Yeah.
Martin, thanks for a great talk. I want to just say thank you for the work you're doing. I mean, it feels like you're sort of a guardrail. And really, you know, I had this statement in my slides yesterday about uncontrolled variation being the enemy of quality, right? There's so many variables that we're missing, and you know, sort of speculating about, and all the clinical impacts you point to. This is just such needed work, and clearly you've laid bare all these problems we're facing. So I guess my question for you is, you you hinted at mass spec, and I'm not a technician, so I'm curious from a workflow, practicality, expense standpoint. How do you see if mass spec does become ready for prime time, the ability to analyze samples and material faster, cheaper? Does that really help us get to where we need to get? It's not the same thing as a high sensitivity ELISA, but maybe it's good enough that it will start to help us evaluate things better.
Yeah, no, thanks, Brian. It's a good question. The issues with mass spec are, number one, you got to lay out 400 to $1 million to buy the instrument at present. And it's actually funny, a few years ago, Peter Breeder and I wrote a review about allergen standardization and testing. And at that time, we predicted that mass spec would need to come down to an instrument that would actually fit on a bench before it could be widely adopted. And that's the instrument that Thermo Fisher produced, the Q Exactive. Because before then, it was actually a whole room full of gadgets. So mass spec has come a long way, but the food industry is going for mass spec because if you're looking for an extensively processed product and you just want to look for peptides that are allergen related, then that's a good technique to use because immunoassays won't usually work in that context. We're using it, as I mentioned, for allergens for which we don't have an assay, and we're planning to run some more detailed studies of peanut later this year where we're comparing different peanut materials. The real challenge for mass spec, which we talked about at the Paul Ehrlich meeting, is there have been no ring trials of mass spec. And so when you look at it, it's not only different instruments, so there's a Thermo Fisher instrument, there's a Shimadzu instrument, there's a Waters instrument, they all use different protocols, and nobody's really done a satisfactory ring trial to show that those are effective. So when we run assays, for example, for the EDQM, we have a couple of assays for pollen allergens in the European Pharmacopoeia. Those all go through a ring trial with all the competent authorities in Europe, and there's probably a dozen labs involved who are making those measurements, and those data are all published. We don't have anything like that for mass spec for allergens, and that needs to be a priority. Having said that, it depends on your application. The other thing about mass spec is you can multiplex it. You can put 10 or 20 samples into the mass spec and it will run them overnight, and the next day you've got this huge data set. And so I think the FDA wants to use it in terms of as an alternative almost to HPLC, where you get a lot of peaks, and if those peaks are consistent, then that's going to satisfy them. I mean, obviously, if they'd been using that for the ALK materials, then we've saved a lot of problems. But also, I think that, again, running these things in clinical trials is that, do you want to spend $10 million on a clinical trial and then discover you have a problem with the extract? Or do you want to start, as Aimmune did, by making the measurements?